ASRB NET 2008 first question paper. So first question paper in the sense in that particular year it is the first time that happens. I mean in that I think it happens in I think in the month of April I think. So ASRB NET plant pathology. These all questions are memory based question papers. For the first time in my while I was studying my first year MSc, so I was first time I attended this 2018 one question paper. Actually I failed for the first time. So anyway, that was a great experience for me. My name is N.H. Shankar Reddy and I am doing PhD plant pathology in Normal University. So coming to the questions that has seen in 2018 one is the first question is agar agar is produced from. So agar agar is produced from Gelidium species, that is red algae. The second one is stem gall of coriander is caused by. So the stem gall of coriander is caused by Protomyces macrosporus, the causal organism. I think spelling mistake here, not macryosporus, it's a macrosporus. So the third one is bud eye spot with the red margins is the characteristic symptoms of bud eye spot with the red margins is the characters of grapes anthracnose. We can see so anthracnose, grapes anthracnose, the character symptoms of grape anthracnose is formation of or production of a bud eye spot. So that is the characteristic symptoms. So the next question is in bacteria 70th type of ribosomes is present in cytoplasm, whereas in bacteria the 70th type of ribosomes can be observed in cytoplasm. So the next question is who reported first time tobacco necrosis satellite virus. So the tobacco necrosis satellite virus was first reported by Cassanis in the year of 1962. Broom grape is an obligate parasite on which crop? So broom grape is an obligate parasite. Actually, the broom grape is normally called as striga is normally called as broom grape, but I don't know whether which crop has act as a parasite on which crop. I don't know if you know. Just comment below. And the next one is pierced disease of grapes is caused by Xylella fastidiosa. Here small L not caps. And pierced disease of grapes is caused by Xylella fastidiosa, and it is the first plant pathogenic bacteria to be completely genome sequenced. So they will ask like this also, what is the first plant pathogenic bacteria to be completely genome sequenced? That is Xylella fastidiosa, that was sequenced by A.J.G. Simpson in the year of 2000. So this first plant pathogenic bacteria Xylella fastidiosa, which was sequenced by A.J.G. Simpson in the year of 2000 with the help of Brazilian consortium. You don't need to remember all those things. It is the first plant pathogenic bacteria to be completely genome sequenced that causes pierced disease in grapes. The next one is shape of growth curve of bacteria, bimodal curve. Not only bacterial growth curve, but also polyatic disease growth curve is also bimodal curve. Next one is viroid consist of. They're asking the genome of viroid. So genome of viroid is single stranded RNA. So far 40 viroids has been discovered. So all the 40 viroids whose genome is composed of single stranded RNA only, not DNA, composed single stranded RNA. So the next one is fungicide with both acropetal, that is upside, downside, that is basipetal movement. So the fungicide which having both acropetal and basipetal movement is phosphatyl aluminum. And the second one is metaloxyl. So phosphatyl aluminum and metaloxyl. You can also better to go with trade names. I think phosphatyl aluminum trade name is Aleti, maybe metaloxyl redomil or subdue, and some other trade names also will be there, but the most important trade names are redomil and subdue. So fungicide with both acropetal and basipetal movement, that is phosphatyl aluminum and metaloxyl. The next one is strobilurin interferes with. The strobilurin group of fungicides are the new generation fungicides. Now I don't think that's now at present I can't call it as a new generation fungicide because where the strobilurin group of fungicides were introduced in the market in 2006. Now we are in 2021, almost 21 and 22. So if you see this, it's been almost it become old generation because it's been almost 14-15 years back. So the strobilurin group of fungicides are actually isolated from a mushroom that is called Strobilurus tenacellus. It is a mushroom where all the strobilurin group of fungicides are isolated from. So here they are asking the mode of action of strobilurin. Simply they are telling, they are asking like strobilurin interfere with. Simply they are asking mode of action only. So the strobilurin group of fungicides interfere with inhibits electron transfer in mitochondria as well as disrupting metabolism. Inhibiting electron transport in mitochondria as well as disrupting metabolism. So the next question is food reserve of bacteria is simply food storage organ of bacteria. We can also call it as food storage organ of bacteria, that is polybeta hydroxybutyrate is the food storage organ of bacteria. So the next one is, as I told you earlier, what is the first bacterium that is completely genome sequenced? Not first bacterium, first plant pathogenic bacterium completely genome sequenced. So the first plant pathogenic bacterium that is completely genome sequenced is Xylella fastidiosa, that was sequenced by Simpson in the year of 2000. I think in earlier slide we had discussed. So the next one is mode of action of triazole group of fungicides. So the mode of action of triazole group of fungicides are inhibits sterol biosynthesis. The next one is virus strain used in cross production is mild strains, because mild strains are used in cross production to provide protection against severe strains of plant viruses. So the next one is time period between inoculation and infection. The time period or the time gap between inoculation and infection is known as incubation period. The next one is blister blight of tea is caused by, we know very well that blister blight of tea is caused by Exobasidium vexans. So the next one is organomercural fungicide. Organomercural fungicide is Serasan and Serasan wet. So among the given options, Serasan and Serasan wet seems to be the reliable one, because remaining options are like carbendazim, mancozeb. Those are not at all a mercury fungicides. We know very well. So Serasan and Serasan wet are the organomercural fungicides. So the next question is virus that infects gram negative bacteria is known as bacteriophage. Not only they want to confuse us, that's why they mentioned gram negative bacteria. Whatever the bacteria that may be gram negative or gram positive, if a virus is infecting bacteria, that is a bacteriophage. That's why they named as bacteriophage. Phage in the sense eater, bacteriophage, bacterial eater. In the sense, a virus that infecting bacteria is known as bacteriophages. Hope you will know that who discovered the bacteriophage. So the bacteriophages were first discovered by Twat and Herrelli in the year of 1915 and 1917. So they both individually discovered bacteriophages. So next one is grape fan leaf virus is transmitted by. So grape fan leaf virus is transmitted by a nematode, that is Ziphinema index. So the next question, black heart of potato is caused by. So black heart of potato is oxygen deficiency or low level of oxygen which is responsible for black heart of potato. So the next one is father of molecular plant pathology. So Noel Keane is considered as father of molecular plant pathology. He is also introduced the term elicitor. The term elicitor was also introduced by Noel Keane. He is also considered as father of molecular plant pathology. So the next one, most sensitive technique for the detection of plant pathogens. So ELISA, among the techniques that we have so far, the techniques that has discovered so far, ELISA is the most sensitive technique that is used to discover plant pathogens, especially plant viruses. So the sensitivity of ELISA is 10 nanogram per ml, whereas the sensitivity of PCR is 10 picogram per ml. So the next one is apple proliferation is caused by. So apple proliferation is caused by phytoplasma. Apple proliferation, which is a phytoplasmal disease. So the next one is the first fungus that is completely genome sequenced. Actually, the first plant pathogenic fungus that is completely genome sequenced is Magnoporthe gracia. It was sequenced by D. Natal in 2005. It's a very, very important one. First plant pathogenic fungus that is completely genome sequenced is Magnoporthe gracia, that is Pyricularia virus, rice blast pathogen simply. So the next one is fungicide applied prior to fungal infection is known as protectant fungicide. A fungicide which are applied prior to infection is protectant fungicide. So the pore size of laminar airflow chamber is 0.3 micrometer. Here HEPA filters are normally used in laminar airflow chamber, we know very well. So the HEPA filter, in a HEPA filter, the airflow chamber pore size is 0.3 micrometer in diameter. So the next one is Agrobacterium radiobacter, which is active against So the Agrobacterium radiobacter, which is active against Agrobacterium tumefaciens. Actually, it is a biocontrol agent which is effective against close related strain, that is Agrobacterium tumefaciens, which causes galls, tumors, and various types of symptoms and disorders in various plants, which was controlled by Agrobacterium radiobacter, that is K84. So basidiospores of smut fungi, I don't know what it means. If you know, just comment below. The next one is Prunus necrotic ring spot virus is transmitted by. So Prunus necrotic ring spot mainly transmitted by pollen, mainly transmitted by pollen. Occasionally it can also transmit by means of sweets, but the first preference goes to pollen only. Prunus necrotic ring spot virus is transmitted by pollen. So coming to the next one, xanthomonas oxonopodis pathovar punicae, not tunicae, panthovar punicae, which causes a bacterial blight of pomegranate. They're asking, they had given the causal organism. They're asking what is the response, what is the disease that is responsible for. So xanthomonas oxonopodis pathovar punicae, which is responsible for bacterial blight of pomegranate. Spelling mistakes will be there, just neglect it. And the next one is systemic acquired resistance induced plants or the external application of. So the systemic acquired resistance, external application of salicylic acid will be required for resistance, whereas in induced systemic resistance, ethylene and jasmonic acid can be applied. So here is systemic acquired resistance, that is salicylic acid, induced systemic resistance, that is jasmonic acid and ethylene. The next one is white halo around the brown spot of tomato. It seems to be blossom introt of tomato, which is a calcium deficiency, but I'm not sure whether it is the symptoms are same or resembles to that same white halo around the brown spot. I'm not sure. So next one is epidemic is for. So epidemic is for late blight of tomato and potato. Sorry, it's not late blight. It's early blight, not late blight. Epidemic is meant for early blight of potato and tomato. So the next question is Cuscuta is a. So Cuscuta is a total stem parasite, whereas Loranthus is a partial stem parasite. Cuscuta, Loranthus, Orabanki is a complete root parasite, whereas Striga is a partial root parasite. So the next one, whitefly transmitted viruses are comes under group. So all whitefly transmitted viruses are comes under group Gemini virus. Next one is fungicide used as a plant defense activator. They had given 4 fungicide, propiconazole or carbendazim, or some of the fungicides has been given. I think propiconazole and as well as benzimidazole also comes under this plant defense activator. I don't think that benzimidazole option will be there or not. I am not sure. I think it seems to be propiconazole will be reliable. If benzimidazole will be there, it also acts as a plant defense activator. So the next one is Indian stem rust rules was written by. So Indian stem rust rules was written by Nagarajan and Singh, whereas Puccinia pathway was written by Nagarajan and Joshi. So Indian stem rust rules was Nagarajan and Singh 1975, whereas Puccinia pathway Nagarajan and Joshi in the year of 1980. So the next one is papaya ring spot virus is transmitted by aphids. So the next one is intracellular PGPR. They had given some options: Bradorhizobium, Azorhizobium, some of the Rhizobium they had given. But intracellular rhizobium, that is Bradorhizobium, is the best example for intracellular PGPR. So the next one is monograph of Fusarium was written by. So the monograph of Fusarium was written by C. Booth. So the next one is indicator plant for citrus greening. So the indicator plant for citrus greening is sweet orange. So the next one is root stalks against collar rot. I don't know. These are all the options that they had given. I don't know the option. If you know, just comment below. So the next one is pathogenic growth and disease can be estimated by. I don't know the answer. If you know, just comment below. The next one is cosmetic fungicide. Example of cosmetic fungicide they are asking. Tridamorph. Tridamorph is an example of cosmetic fungicide, whereas rescue treatment fungicide, that is triadimifon. Triadimifon. So you can also go with trade names. I think the trade name of Tridamorph is Calixin, whereas the trade name of rescue treatment fungicide, that is triadimifon, that is Bileton. So I am coming again. Cosmetic fungicide, example of among the given options, Tridomorph is the cosmetic fungicide. Trade name is Calixin, whereas Rescue treatment fungicide example is triadimifon. Trade name is Baileton. So these are all the actually these questions has been sorry, these questions are not asked in a net question paper. For your understanding, I was placed here. So indicator plant for citrus tristiza virus is Claxylem. I think spelling might not be correct. You just check it out. And citrus tristiza virus is caused by indicator plant is Claxylem. Citrus greening indicator plant is sweet orange. Whereas citrus exocortis, not exocortosis, exocortis. So exocortis indicator plant is trifoliate orange. So phytoplasma indicator plant is periwinkle. These are all some of the indicator plants. So the next one, oh yes, here we can see here the rescue treatment fungicide, which is an example of rescue treatment fungicide. That is triadimifon. Trade name is Baileton. Baileton is the trade name of triadimifon. The next one is MOA, in the sense mode of action of carbendazim. So the mode of action of carbendazim, that is disrupts cell wall. So the next one is Keira disease of rice can be controlled by, we know that Keira disease of rice is due to zinc deficiency. So if it is by zinc deficiency, if we apply zinc, we can control the disease. That's why they are giving application of zinc sulfate can control this Keira disease. So the next one is water preservation of a fungal culture was suggested by. So fungi can be preserved in water, that can be suggested by Crystallania. Crystallania is a scientist who suggested that water can be act as, or water can be used as the preservation of fungal cultures. So the next one is 14 opines are produced during a pathogenesis of agrobacterium. We can see 14 opines. I think not only 14, more than that, I think 21 opines are there. I think I'm not sure, but 14 opines are can be observed during the pathogenesis of agrobacterium tumefaciens, simply agrobacterium. So the next question is phytoplasma infected phloem cells during staining. The phytoplasma infected phloem cells will give blue color. It's a very important one. So the phytoplasma infected phloem cells while staining, that will use blue color. Blue color. They had asked what is the color during staining of phytoplasma infected cells that we can observe. That is a blue color. Define vertifolia effect. Vertifolia effect in the sense loss of horizontal resistance while breeding for vertical resistance is known as vertifolia effect. The concept was given by Van der Planck. So the phytoplasma is sensitive to tetracycline. Actually the spelling is wrong. So it is tetracycline. So phytoplasma is sensitive to tetracycline but resistant to penicillin. Next one is hump-beaked iridospore, not humped-beaked iridospore. Hump-beaked iridospore is produced by Hemilia. Next one is we can also see turnip-shaped teliospore, hump-beaked iridospore, or kidney-shaped iridospore, which is produced by Hemilia only. Hump-beaked or kidney-shaped iridospore, turnip-shaped teliospore, both are produced by Hemilia only. The next one, in PCR technique, which is antibody used? They're asking in which PCR technique antibody used. I don't know the answer. If you know, just comment below. The next one is formation of pinwheel, not pinwheel. Formation of pinwheel in cytoplasm is the characteristic symptoms of which group of virus? That is potivirus. The formation of pinwheel inclusion bodies in the cytoplasm is the characteristic feature of potivirus. RTV contains rice tumor virus contains two types of particles, that is rice tumor spherical form virus and rice tumor bacilliform virus. I think that is if there is it in options, I think that is the correct one. So the next one is Withing syndrome or Whiting syndrome with a crescent phase. Simply for confusing us, they are adding something like Withing or Whiting syndrome, something like that. But the crescent phase, if we see the crescent phase, we can immediately observe, we can immediately remember that the bacterial blight of rice, that is no, in that particular bacterial blight of rice disease only we can observe this destructive phase, that is crescent phase. So the next one is angular leaf spot of cotton can survive in seeds. Angular leaf spot of cotton can survive in seeds. So the next one is first record plant pathogen in the world, that is giant mosquito or loranthus. It is the first plant pathogen that is recorded in the world, which was first observed by Albertus Magnus in 12th century. So that is giant mosquito or Lolanthus. So the next one is Cleistothesia, that is dichotomously branched Cleistothesia with a single ascus. So dichotomously branched Cleistothesia with single ascus, that is Podospara. If it is a Cleistothesia dichotomously branched with many ascus, if it is a single ascus, that is Podospara. If it is a many ascus, that is a Microspara. So the next one is hot ignats are found in ectomycorrhizae, whereas in endomycorrhizae hot ignats are absent. The second one, next one is ICTV. The year of establishment of ICTV is the 1973, whereas ICNV is the 1966. The few of some of the story that I have to tell regarding this. ICNV in the sense International Committee on Nomenclature of Virus, which was established in the year of 1966, which is a chief governing body that can name and that can give taxonomic position, all those things for plant viruses. Whereas the same ICNV, International Committee on Nomenclature of Virus, which was established in this 1966, that is renamed as ICTV, that is International Committee on Taxonomy of Virus, in the year of 1973. ICNV, Nomenclature of Virus, 1966. ICTV, Taxonomy of Virus, 1973. So the next one is endospore forming bacterial genus. So the Bacillus and Clostridium are the two genus that can form endospores. I think Clostridium was not there in the options. For your understanding and remembrance, that I had mentioned one more thing extra. So the Bacillus, which is a gram Bacillus, and Clostridium, both are the gram positive bacteria that can produce endospore. Hope you will know about endospore, which is a heat resistant spore that can be that can use to survive in harsh environmental conditions. And the next one is exclusion by legislation is known as plant quarantine. So here the next question is which race causes Moko wilt disease? We know very well that Moko wilt is caused by Ralstonia solanacearum. Here in Ralstonia solanacearum there are 5 races that can affect different types of crop plants. First, race 1, race 2, race 3, race 4, race 5. Race 1 will affect Solanaceae. Race 2 affect Musaceae. Race 3 affect potato. Race 4 affect ginger. Race 5 affect mulberry. So if they will ask something like that, we can easily identify that. So Moko wilt of banana. Moko wilt is a disease that can affect banana. So that is Musaceae, the family of banana. So that is race 2. They are asking. So Moko wilt disease that can affect which race? Race 2, Musaceae. Race 1 is Solanaceae in general. So if they ask about particularly potato, that is a race 3. Race 4 is ginger. Race 5 is mulberry. So the next one is leaf wetness period of apple scab. So the leaf wetness period of apple scab is 9 hours. You can see apple scab. Totally 9 letters are there. So 9 hours. You can remember like this also. So the next one is which crop is the most sensitive to ozone damage? Which is not sensitive to ozone damage. If it is a if they had asked which crop is the most sensitive for ozone damage, tobacco is the most sensitive for ozone damage. But they had asked which crop is not sensitive to ozone damage, but I am not sure. Maybe tomato, might be tomato, which is not sensitive to ozone damage. So I'm not sure, that's why I never mentioned the answer. If you know, just comment below. And the next one is autoclave temperature at 15 lb for inch square for 30 minutes, that is 121.6 degrees centigrade. So the next one is syringomycin is an antibiotic only. So the next one is family of Irwinia. Family of Irwinia is Enterobacteriaceae. So the next one is ratoon stunting. Ratoon stunting of sugarcane is caused by Lifesonia xylem subspecies xylem. It is the new name where it is previously called as Clavibacter xylem subspecies xylem is the previous name of sugarcane ratoon stunting. Now it is called as Lifesonia xylem subspecies xylem. It transmits through infected sets. So the next one is stage absent in the demicyclic rust. Demicyclic rust name itself indicates lack of iridial stage is considered as demicyclic rust. If there is no iridial stage, it is called as demicyclic rust. So the next one is tumor-inducing principle of crown gall was discovered by. So the tumor-inducing principle of crown gall was discovered by New and Kerr. These are the scientists who discovered the crown gall principle, which is a tumor-inducing one. The next one is symptoms of downy mildew of pearl millet. I don't know, different types of symptoms has been given, but at that time I don't know the exact symptoms. So the next one is xanthomonas axonopodis pathovar punicae. It's not capital, it's a small one. Xanthomonas axonopodis. Xanthomonas axonopodis pathovar punicae. It affects all the parts that has given. It affects leaves, it affects fruits, it affects stems. So if I want to point out a exact or specific one, fruits, because fruits are the economic points where cracking, LR shaped cracking can be seen on fruits. And one more thing, the symptoms can also be observed on the leaf and on stem, but they had given options like leaf, stem, fruits, and in the last option is all the above. So the answer is all the above. So the next one is citrus gummosis is caused by. So citrus gummosis is caused by Phytophthora citropthora. Option is they had given this option. Actually, this citrus gummosis is caused by 6 species of Phytophthora. Phytophthora citropthora, Phytophthora parasitica, Phytophthora palmivora, Phytophthora syringae, Phytophthora, and some other 2 species are there. Actually, I didn't remember. But among the given options, Phytophthora citropthora is the reliable one. So the next one is conidial germination for Pyricularia varizae requires how much amount of relative humidity? They are asking. For the conidial germination of Pyricularia varizae, it requires 90 to 95% of the relative humidity will be required for the germination of conidia. So V-shaped chlorotic lesions are the characteristics features of. Spelling mistakes will be there, don't consider it. So V-shaped chlorotic lesions are the characteristic symptoms of black rot of cabbage. In black rot of cabbage we can see the V-shaped chlorotic spots. So the next one is watery soft rot of carrot or soft rot of carrot is caused by Irwinia carotovora, pathover carotovora. We know very well about this. So the next one is dyes used in gram staining. So dyes used in gram staining is methylene blue. Different types of dyes can be used for different types of purposes. If you take saffron, that is a counter stain or counter staining technique. I mean, for counter stain, saffron can be used. So for different chemicals can be used. In general, methylene blue can be used as a dye in gram staining. So the next one is cotton rust is caused by. So cotton rust is which is a potassium deficiency. While I was writing, I don't me too don't know the answer. Just I know I was refreshed it from my friends and my seniors too. So cotton rust is caused by potassium deficiency. I thought it is a fungal disease, something like that, but it's not true. It's a potassium deficiency. So the next one, because the name, if you see, if you listen this, the name itself rust, we thought that it may be Puccinia or some other species of rust genera, but it's not true. It's caused by potassium deficiency. So the next one is foot cells, comma vesicles and phalloids, not phalloids. Phalloids are present in aspergillus. So food cells, vesicles and phalloids are present in aspergillus. I think they had given some other options like aspergillus or penicillium. Penicillium also consists of food cell, vesicles and phalloids, but I think penicillium will not be there in that particular option. Even though if there, the preference will goes to aspergillus only. So the next one is critical stage for Taphina deformans. If one spray remains, I don't know the answer. If you know, just comment below. So the next one is late blight of potato in Ireland is due to wherever it is a late blight of potato, that is due to Phytophthora infestans. It may be late blight, it may be in Ireland or it may be in America, it may be in India. If it is a late blight, it is caused by Phytophthora infestans. So the next one is oyster mushroom. Which of the following species is oyster mushroom? I think they had asked like that. The Pleurotus, Pleu, Pleurotus sozarkazu is the oyster mushroom. The next one is blossom end rot of tomato. Blossom end rot of tomato is caused by calcium deficiency. So the next one is gram positive bacteria contains how much amount of mucopeptides? So gram positive bacteria contains 85% of mucopeptides or 85% of peptidoglycan, whereas gram negative bacteria contains 3 to 12% of mucopeptides, whereas gram positive bacteria contains 85%, whereas gram negative is 3 to 12%. So gram negative bacteria is lack of. So gram negative bacteria is lack of teichoic acid, whereas gram positive bacteria consists of teichoic acid. So the next one is a multiplication of viroids is with the help of enzyme, that is RNA polymerase 2 is the enzyme which is responsible for the multiplication of viroid. If it is a virus RNA polymerase 1. RNA polymerase 1 is responsible for the replication, enzyme responsible for the replication of virus, whereas RNA polymerase 2 is the enzyme responsible for the replication of viroid. Okay. So the next one is, which spore is haploid? I think they had given options like oospore, zygospore, or chlamydospore, something like that they had given. So which spore is a sexual haploid spore? So chlamydospore is not at all a sexual spore. So the answer goes to ascospore only, because ascospore is a haploid sexual spore. We know very well about ascospores and basidiospores are haploid sexual spores, whereas zoospore and zygospores are the diploid sexual spores. The next one is, bacterial respiration is increased. If a bacterium which is enters into the plant cell, the bacterial respiration will be increased. The reason is due to binding and unbinding of oxidative phosphorylation. The bacterial respiration will be increased initially when it enters into the plant cells. So the next one is, budding spores are called. So budding spores are called blastospores. So the next one is, the disease dispersed by water splash. I think I don't know what are the options that I had given. If we see at right now, I might be answer. At those time I don't know what is the disease that is dispersed by water splash. And the next one is, if you know, just comment below. The bacteria, that is question number 94. The next one is, bacteria that affect another bacteria is called Dello Vibrio. So Dello Vibrio was discovered by Slope. Dello Vibrio was discovered by STOLP, Slope, in the year of 1962. Whereas bacteriophage, we know very well what is bacteriophage. Bacterial eater, in the sense virus that infect bacteria, that is called bacteriophage, which was discovered by Twat and Herrelli in the year of 1915 and 17 individually. Whereas whereas a Dello Vibrio, in the sense a bacteria that infect another bacteria, that is Dello Vibrio. So the next one is, first cloned plant toxin. So tab toxin is the first cloned toxin that is isolated from Pseudomonas syringae pathovar tabaci. But the first cloned plant toxin is tab toxin, whereas first purified toxin is Victorin, which is isolated from Helminthosporium victoriae. So the next one is AQ10. AQ10, in the sense Ampeliomyces or Ampelomyces quisqualistin, which is used as a biocontrol agent against. We know very well that AQ10 is the specific biocontrol agent that is recommended for grapes powdery mildew. So the next one is a mutant that not synthesize essential metabolites. These are all the options that are given. I don't know the answer. If you know, just comment below. And hypersensitive reaction occurs in, I think, locules or vacuoles, something like that, I'm not sure. So the next one is a bunch of flagella at both ends. So they're asking flagellar conditions. In Lopotrichus, we can see the bunch of flagella on both ends. In Cephalotrichus, we can see the bunch of flagella or flagella at only one end only. In Lopotrichus, we can see the bunch of flagella on both ends. So the next one is indicator plant used for the potato spindle tuber viroid. So the indicator plant that is used for potato spindle tuber viroid is tomato. So the next one is RT-PCR used for the detection of. So RT-PCR used for the detection of virus, viroid, viruliferous vector. Answer is all the above. RT-PCR can be used to detect virus, viroids, and viruliferous vector, all the above. So the next one is technique to produce virus-free plant. We know very well that meristem tip culture can be used to produce virus-free plants, because the meristematic tissues are the continuous multiplication or generating tissues where the virus can't able to live in. So this meristem tip culture can be employed in the production of virus-free plants. So the next one is first empirical mycologist in India in 1905. So E.J. Butler is the first empirical mycologist in India in the year of 1905. So next one is white tip panicle of rice is caused by. So white tip panicle of rice is caused by a nematode, that is Ephilingoides or Ephilingoides bessi. So the next one is L form of bacteria means, L form means cell wall lacking bacteria. L in the sense Lister Institute, where it was discovered by Kleine Nobel. She is a scientist where she worked in Lister Institute. So to honor the Lister Institute, she named it as a L form bacteria, that is a Lister form bacteria, in the sense cell wall lacking bacteria. So the next one is PR proteins are accumulated in apoplast and vacuoles. I think I had said it in for hypersensitivity reaction. It's not true. So PR proteins can accumulated in apoplast and vacuoles. So the next one is greenish growth at the lower surface can be might be initiation. I'm not sure. If you know, just comment below. The next one is a buckeye spot of tomato first infects. I don't know the answer. The next one is foot rot of lilium is caused by two. I don't know answer. So the next one is yellowish translucent or translucent spots or translucent spots are the characteristic symptoms of, I don't know. If you know, just comment below. The first A virulent gene is discovered by Staskowitz. First A virulent, it is a bacterial A virulent gene which was discovered by Staskowitz. If it is a fungal A virulent gene, it was isolated by DeWitt in the year of 1991. Second one is next one is most of the plant pathogenic bacteria are gram negative. Most of the plant pathogenic bacteria are gram negative except some bacteria, that is you can remember BSC. So from BSC only all will come. No, B stands for Bacillus, S stands for Streptomyces, C stands for Clostridium or Corynebacterium. These are all the gram positive bacteria, except that remaining all are gram negative bacteria. So Candidatus, not dates, Candidatus phytoplasma, which is a less Candidatus phytoplasma less non-helical that colonize phloem cells and insects, that was given by, I don't know, even just comment below. The next one is bitter pit of apple is due to imbalanced water supply. So bitter pit of apple is mainly due to imbalanced water supply. So the next one is taro blight of colocasia resistant variety. These are all the options given. If you know, comment below. The next one is single celled light colored conidia. So single celled light colored conidia is produced by cephalosporium. So the next one is contagium vivum fluidum was given by Bizernick. Contagium vivum fluidum was given by Bizernick, and he is also coined the term virus. He is considered as father of plant virology, we know very well. So the next one is muriform septations can be found in alternaria. It is a very important one. Muriform septations or alternate and vertical septations can be observed in alternaria. So if you see the conidia, we can see like this muriform septations like this, and some of the sept are horizontal and vertically horizontal and vertical septations can be seen. So the next one is first line plant defense mechanism. So in plant defense mechanism, the options given is thick cell wall, phenolic compounds and proteins are some other given. So if you take the first line in plant defense mechanism is a thick cell wall is the first line in the plant defense mechanism. So the next question is chemical used to control black tip of mango. So chemical used to control black tip of mango is application of borax, that we can control the black tip of mango. So the next one is loose smut is a, we know very well that loose smut is a internally seed-borne, is a best example of internally seed-borne pathogen. And the next one is mycovirus, genome of mycoviruses. The genome of mycovirus is double-stranded RNA. So the next one is Phytophthora infestans survive as, under unfavorable conditions, the Phytophthora infestans can survived in infected tubers. Under unfavorable condition, under off seasons, the Phytophthora infestans can survive in infected tubers.